All right, in this video, we'll begin Unit 3, which is going to be cellular energetics, which is going to deal with the topics of enzymes and cellular energy in general. And specifically on cellular energy, we'll be looking at the processes of photosynthesis and cellular respiration as they kind of encompass metabolism for most living things. And so we will talk about that. In this unit, we'll begin by talking about enzymes. So a couple things about enzymes. Enzymes are biological catalysts. We'll talk more about that in 3.2 as to what that means. But in general, a biological catalyst speeds up chemical reactions. Most enzymes are proteins, and their shape determines their function, just like any kind of protein does. The tertiary shape, if you remember the primary, secondary, tertiary shapes, the tertiary shape of that protein has to be maintained for the enzyme to have functionality. And so if you remember, when we talked about proteins, there are certain things that can denature proteins. We will go more into that in a later video also. A couple things specifically: there's a region of every enzyme called the active site. The active site is where the reaction is actually taking place, and the substrate is the reactant in this reaction. So the enzyme is doing the work, the substrate is the thing that is being worked on, if you want to think of it that way. The substrate is the thing that is being changed in this reaction, and the enzyme is doing the changing. It is the tool that is doing that. The enzyme, after the reaction, is unchanged. It is something that will stay the same. It can actually do this reaction multiple times. And so the substrate is what's going to change. Let's talk a little bit more about the active site. The active site specifically interacts with the substrate. It is not just any active site. It is an active site specifically for this substrate in this particular example. If you want to go back to the previous one, notice this active site only fits this substrate. So it's very specific. The fit is not exact, and what we call it an induced fit. There is a physical and chemical compatibility. Physical in that it actually physically fits. Chemical in that there are chemical reactions occurring along the ridge or the edge here of this substrate and enzyme, like temporary bonding sorts of things, typically hydrogen bonding, van der Waals forces that are going to temporarily attach these things so the business can occur. The active site will have kind of an induced fit around the substrate. You can see here, it's not exactly shaped that way, but once the substrate binds, you have this nice clean fit. I like to think of it as like a rubber glove. The rubber glove is not specifically designed for a particular hand, but any hand can fit in it, and it kind of binds around the hand in some ways. And so think of it as like an induced fit. This ES complex here is the enzyme substrate complex. Sometimes you'll see that terminology uh, when it's referring to the entire thing, the enzyme and the substrate bound together. Enzymes, the name of an enzyme is often indicated by the substrate or the chemicals that are involved, and most enzymes will often end in this ASE suffix. There are obviously exceptions to that, but anytime you see this ASE, it is telling you something. It's telling you that this is an enzyme, and typically the first part is telling you this is what it does. Like lipase or protease. These are breaking down lipids or proteins. Um, phosphoexoisomerase is an isomer, it's changing the shape of something. Um, decarboxylase probably removes a carboxyl, right? Dehydrogenase, I think you're getting the idea. Some of these are a lot more specific so knowing some Latin might be helpful. But you're not going to have to look at a name of an enzyme and tell me what it does unless I specifically tell you. But knowing that ASE is an enzyme is a helpful thing. So enzymes, just talking a little bit about the way that enzymes react. First of all, I mentioned earlier that enzymes are reusable. The substrate is what's changed. Enzymes can be used again. Depending upon a cell's function, a cell will maintain a certain level of enzyme to deal with the substrate at hand. And what this picture is showing you, that the enzyme will only react as long as there is substrate available. the enzyme's not doing work if there's no work to be done. Once all the substrate is gone, the enzyme will kind of taper, or the reaction rate will kind of taper off. So as long as there's substrate available, it's going to go up. But when the substrate availability decreases, the enzyme activity is also going to decrease, which is exactly what you would expect. In general, the two kinds of reactions that are occurring with enzymes are going to be synthesis or digestion reactions, is the exact language used in the AP course outline. But we can also think about these as anabolic or catabolic reactions, whereas anabolic reactions are building things up. This would be synthesis, and catabolic reactions are breaking things down or digesting things. And so think about it in those two ways. Enzymes are typically either building or breaking things apart.